So first, we just wanted to ask, like, why do we save seeds? And this is an invitation to you all, just in the chat. Why do you save seeds? Why are you here today? What does a seed mean to you? Um, and you can take that as literally or as sort of esoteric as you want. And so just as you're introducing yourselves using that chat space, we can use that also as a space to just see who's here and what's really motivating us and what kind of like is we're holding kind of in our hearts as we think through this special role that seeds play. So we just want to ground in the fact that for centuries, our sacred relationship to plants has been completely foundational to the development of our traditions, cosmologies, food, and cultures. And so as plants, like all of these wild relatives in all the plants that we now consume as foods or medicines, they evolved to specific climatic conditions, different regions. They've existed through these long expanses of time. And in all of those experiences, as they encountered these different, stressors, things they responded to, that changed them at this sort of beautiful genetic level, right? And as they sort of developed all these unique responses to environmental factors, humans began to interact with them too. We started responding to like what we translate in those interesting sort of like coding for proteins and different enzymes and the relay of all this information that's happening on their cellular level. We interpret that as things that taste good or are healing or special, just cosmetically striking and beautiful. And those observations and that sort of mirroring of one another stabilizes those new combinations, right? So in a lot of ways, plants are constantly rolling the dice, trying to figure out these different survival mechanisms. And we streamlined the process and became really kind of collaborators and conspirators with these plants by engaging with them in some way that kind of entered into this agreement and this relationship saying, we're going to, essentially keep stewarding these desirable traits for one reason or another. And that in turn stabilizes what are called like, these different branches on phylogenetic plant tree, family trees. And so you can think of the kind of prime example that I think everyone can relate to is, you have wild brassicas growing throughout the Mediterranean that look something like this. And then you have this amazing differentiation, right? So just within one genus and one species, right, you have this incredible amount of diversity, like in terms of just completely different parts of the plant that people selected for in terms of their edibility. So you have all of these distinct, like arguably 6 or 7, like kind of distinct cultivar kind of buckets, all stemming from one common ancestor. And so if a plant can do that just even within one species, like you think about just the incredible amount of diversity, like when you think about cutting across multiple species and interspecific hybrids and things like that. So I always think like for me, a crop that both highlights specificity and really champions like the unique places we all come from, but is also so unifying is the chili pepper, right? So the chili pepper is native to like a really specific range, like from Central into South America. But it has traveled the globe, right? It is like the real gregarious globetrotter of vegetables. And at each place and each people that it met, what I think is so striking is it just became so deeply and rapidly embedded kind of in the culture. So for me as a Korean American, there's such a particular visceral feeling for Koreans when you smell gochugaru, and when you see kind of these moments. And it's that true kind of like transportive ratatouille moment where if you see someone open a bag and smell ground Korean chili peppers, you really kind of pure visceral feeling can wash over you, and you know exactly what that's going to smell like, and it's kind of tied to all of these different things. And I love that the pepper is just such a good example because it's not merely sort of a nationalistic bend of just like kind of claiming something. It really is speaks to the kind of like beauty in that relationship in terms of like a mutual sense of belonging to one another. And those peppers like have this imprint of the people that really started to kind of shape certain profiles based on like all of these different factors. The chili pepper's genome is like incredibly long compared to like its closest other living relative, the tomato. So it's got the same amount of chromosomes, but it's got like more than 4 times the amount of space within those chromosomes functionally to tweak different sort of novel traits and combinations and recombinations. So with all of those opportunities, like you think of all the people that interacted and made a chili pepper feel like it was our own. Um, so I think overall for our time together today, I wanted to just ground us in the fact that when we talk about seed saving, you know, the removal of a seed from a fruit or a pod is like a straightforward process. But we really want to hold like the bigger process that we're talking about with seed keeping, and understand that beyond just like the sense of like this physical resource that we have, um, that's merely a reflection of like all of the time and care and love that people have poured into these plants, and that those plants have really met us with these acts of care and tending to us in terms of shaping our culture and our character as well. Um, so I think we just really want to hold that just because there's just nothing like that, I think, in terms of like being able to hold a seed and feel like you have something that is both anchoring you to all the people and all the places and all those plants that came before, and is also tying you so firmly to the future in terms of what it asks of you, what it asks of you to think as like a thought partner and, strategist to think about our mutual survival, which is what that sort of co-domestication really looks like. So for me, when we talk about seed saving basics, we want to really work from a place that is about that web of relationships, and we want to look at those different facets within that web. So I want to talk about this. If there's one mission with this workshop, it is to sidestep what I call the accidental seed saver. And so a lot of times we hear friends just, walking around their farm, looking in their garden. There's the overgrown zucchini in some corner of neglect. And it's like everybody can relate. The zucchini patch gets away with you without fail every single summer, right? Like you just cannot keep up at a certain point. And so when I hear people being like, oh, that's, the zucchini I missed at the end of the season as things are winding down, like, oh, all these zucchini that we are left over from when we stopped harvesting. Well, sure, we'll just save those for seed. And that's great. And functionally, yes, you can get a seed from that fruit. But what we want to explore today is to really optimize the role that seed saving can play, like on your farm or in your garden, for a number of different things and outcomes. You really want to start every single season centering seed in all of your processes and all of your design. So we never want seed saving to be the afterthought of the thing that we missed, the thing that happens at the end of the season when we're, when we're tired of eating zucchini, then we just let it peter out and we save the seeds, right? Because that's all well and good, and you can grow seed from that from year to year, but you're missing like what the really beautiful kernel of opportunity and that really expansive opportunity is, which is just to think really mindfully about all the things that you're preserving and all the opportunities to have seed that is, again, really so finely attuned to like your climate, your region, the specific stressors on whatever little patch of land that you're on. And also thinking about just the creative parts of this, like, right, like to acknowledge like all the different things that the plant is trying to figure out from season to season, and how you like really engage with that, and again kind of work in tandem with it. And that give and take is just such a beautiful part of our partnership with plants. So we want to just kind of talk a little bit about this. I think with a lot of workshops, people just think, yeah, like, oh, I thought we were going to be threshing and winnowing and whacking stuff with sticks, and now you're just talking about basically most of the time we're going to spend together is more about using a pen and a piece of paper, taking pictures and making observations. But all of that is really, like, to me, the work of seed keeping. Extracting a seed from, again, like the vessel that it's contained within, is just like really an end product, and really just a really tiny percentage of like the true work that it takes to do this seed saving work. So the jumbo zucchini is the image we want to keep in our mind. Like, okay, we can do better. So we try to break things down when we talk about designing seed projects. We think about it almost as in just like the physiological processes of a seed emerging from the soil, right? So you think first of conditions, right? And a seed is like such a beautiful, complete package in this way. And what we need to provide is so minimal at that very beginning stage, but there are fundamental fixed things that a seed just needs, right? So that's, seeds need sun and they need light and they need water. They need water to imbibe and crack their little seed coat and put out their first little radicle. So we want to think about those things when we start thinking at the beginning of the season of like a crop we want to save their seeds. What is the design of our project? What are fundamental elements needed to support this plant? So these are just the things that, yeah, it's just kind of innate for that plant character. What is their life cycle? What is their pollination habit? What are any sort of reproductive mechanisms needed to actually like make seed? We want to know kind of like different population sizes to keep the plant healthy. And we want to know their basic water and fertility needs. Like what's the baseline of what they kind of like and what they would like to see in order to survive. Next, we want to think about context. And so for us at second generation seeds, like we think a lot about, yeah, again, just what we can see reflected back in like those plants and all the stories that they're carrying with us. And just the fact that so many people, those seeds really are a direct line to these other broader stories that we're telling within our communities. Um, and so with a lot of things, we want to just also understand, like if we think of that seed cracking open and then it's got its cotyledons, right? That's the first thing we start to see and admire above ground. And a cotyledon is like this beautiful reflection of just everything that plant's ancestors passed on, everything the parent plant has equipped that seedling for in that next season. So this is everything that plant knows directly from its parents, right? So it's the blueprint for that plant of all of that genetic information. And so we want to think, what are the cotyledons of our project? What are all the things that we already know and appreciate? What are all the things that are coming through, all these transmissions from past ancestors? We want to think about what are the characteristics that make them unique. And we want to think, are there traits that we want to remain fixed? So what do I like about this variety already? And if I'm going to adapt it for different things, what needs to be there so that I still have that tether, right, to all the things that I like, if I'm going to continually try to improve it for the future. The next thing we want to think about is curiosities, right? And this is where our imagination comes in. So we want to think, now with that plant, imagine it's making its first true leaves and it's expanding. That's when the plant starts to tune in. Hey, where am I right now? What is this like? How long are my days? What is the temperature? What is the soil like? What is the quality of the water? How am I being cared for? So all of these things that start developing in response to that plant, better understanding the world that it's living within. These are our curiosities for the future. So how does this plant interact in my climate, within my farming system? And this gives us clues of things that we need on our end in order to have that plant really become well attuned to our care. So this is where we start to identify areas for improvement or adaptation or breeding. And we want to really think about what are the critical factors that will impact this plant's future. So for us in the Central Valley, we start doing a lot of adaptation projects because, increased salinity in our groundwater, intensity of heat and drought. So we start thinking about, if I want, future generations to enjoy this plant, what are the things that I need to think about and start building into my project, like a curiosity to kind of ensure that I'm being accountable to this plant for future generations. So to elaborate a little bit, conditions designing your project, plant's life cycle, right? You have annuals, biennials, perennials. All of them will follow kind of like, as a flowering plant, like a very broad sort of outline of that life cycle with just different sort of nuances in between, right? In terms of like a perennial is not going to flower on the same schedule as an annual or a biennial, but at the same time, it's going to be producing vegetative mass, moving into reproductive maturity, maturing seeds, and so on and so forth. Pollination habit. Plants are either mostly self-pollinizing or they're outcrossing, right? A lot of flowers might be self-fertile, like the flowers on them are perfect, but their tendency might be, yeah, again, a little more gregarious, a little more open to influence from their peers. So even though something can be mostly selfing, they're still capable of outcrossing, but there's just sort of a spectrum of how those plants behave. And you want to just sort of understand kind of a broad idea of that because it'll inform also your population size. And then the next category, which is isolation distance. And so if you want to maintain a variety in really particular ways, you will want to think about the space that it needs from another variety that you're also trying to maintain. And so pollination habit really guides that. So something like lettuce is extremely self-pollinizing. The risk of crossing is very minimal. Something like soybeans too, the flower is pollinated pretty much before it opens. So in that case, you can grow a lot of varieties practically next to each other. Whereas other crops, like peppers or eggplants, there's a slightly higher chance of outcrossing depending on your climate versus things like squash, which are just really there to mingle and party. And in which case you really need like a pretty big gap between them. One of the other main considerations is reproductive mechanisms. So when plants start to shift from making kind of vegetative growth and this leaf matter in order to produce food to ultimately like have food to pass on and make healthy viable seed, there's a lot of mechanisms that trigger a reproductive maturity, right? And that could just be a certain like literal amount of days that it takes of growing vegetatively. But there's a lot of things that are kind of nuanced, like photoperiodism. So it's day length sensitivity. So you hear short day or long day plants. And those plants really only flower once they sense either the lengthening or shortening of days. So these are things that, really start to come into play. Like perilla is my favorite plant, and it really inhibits like how far north or south. Like it dictates the complete latitudinal range because the flowering won't be activated outside of a certain fluctuation of day length. So those are just things to know, like if you're thinking about an adaptation or a breeding project, it's something like relatively fixed that will be very hard to influence at times. Or vernalization is another example. So with a lot of biennials, the process of vernalization, which is like exposure to cold weathers, in order to trigger again reproductive phase to start happening. And temperature differentials is another factor, although in some ways it affects the entering of reproductive maturity, but in a lot of ways it mostly affects fruit set and successful pollination. And so, with a lot of common beans or even runner beans, there's like the best fruit set and seed development will happen when you have a pretty good gap in terms of the nights cooling down coupled with pretty warm weather during the day. But if you're in places where it stays hot through the whole night, then you might end up with a lot more just flowers aborting and poor fruit set, things like that. The next thing to talk about is population size. And so we're going to get into this in a little bit more detail, but you're looking at like kind of the minimum amount of plants that you need to grow viable seed, to preserve a variety and keep it kind of consistent over time, or to really ensure avoiding genetic bottlenecks or avoiding kind of perpetuating sort of negative or deleterious traits. So long-term viability, and those are three different kind of figures. So like some plants, right, they're self-fertile, they're mostly self-pollinizing. So you could grow one plant and have seed that will germinate and grow. If you're thinking about the long-term health though, you start getting into these bigger populations needed in order to just have enough variation and enough other potential opportunities for kind of novel alleles to develop. And so in that case, you need to start upping your population size when you start thinking about something that is like really beloved. You can't imagine living without it. You need to kind of like really orient yourselves to this idea of growing large enough populations to maintain that. And then water and fertility needs, again, that's just letting you kind of plan out the space on your farm or in your garden to just know like where things are going, where things are mapped out. And you want to just also think about those things because it can inform if you start thinking about long-term breeding projects, like where there might be areas that you can affect. So you want to know what's your baseline, because then you can sort of establish that baseline to try to develop more drought tolerance or more capability of thriving with less inputs. Okay, so when we talk about context now, what are we talking about? So we want to just acknowledge the perspectives or cultural connection that sometimes has shaped that plant. When we think about the characteristics that make them unique. So with us, when we work on any sort of, even if it's just a preservation project, but a lot of times too, when we do cross farms or like multi-farm trials, you want to just start to map out all of these different characteristics into their component parts. And that in plant breeding is called like a phenotyping protocol, but it's really just a map of like you painting a portrait of this plant. So you start with physical traits, like things that are just easy to evaluate and observe just looking through the field, the things that are striking. So it's like looking at leaves, flowers, fruits, and seeds, observing their shape, color, texture, right? Anybody, like you can just do that kind of, that's like your sensory part, just observing. Then you get into kind of like any sort of culinary, medicinal, like sensory evaluations. So that's your flavors, aromas, textures. And then you can think too of usage, which is just a relationship between like ways that that plant has maybe been prepared that influences selection for these other characteristics, right? So, we do like squash trials, and in a lot of breeding programs, the way you evaluate squash is to cut them into cubes and microwave them. And there's seen to be this like objective sensibility, like, oh, we microwaved all of them. And so this tells us we have an objective measure for what's delicious. But I think we just want to encourage like more complex rubrics of who decides like the value and utility of things. So we always try to incorporate a sense of how did different people use these different crops, like something like squash? Was it stewed? Was it turned into soup? Was it roasted? Was it steamed? Because there are things that translate to our selection criteria that we might do a disservice to if we don't acknowledge the ways that it was used. And then again, are there traits that should remain fixed? So what maintains a sense of legibility? So for us, like doing a lot of crops that are important to Asian American communities, it's a question we ask a lot. If I bring this to that community who's historically stewarded it, will it be recognizable? And if we're breeding for new novel traits, what are the things that still feel like it's including the perspectives of that community? And so this is just a resource that people wanted to throw the slide in here so that when people have it after this presentation, you could look it up, because this is a little bit of detail about different traits in vegetables and whether those traits are dominant or recessive. And so when you start getting into preserving things, or if you even see something novel, just like randomly happen in your field, like you end up with a type of lettuce that you really love, it's suddenly like there's a couple heads in the population that have a different color. This can kind of help you understand long-term what you're looking at in terms of really fixing the trait that you like. So it's just kind of a good table to refer to. So lastly, we talk about curiosities, and this is just all the areas that we know we need to deal with, whether we're looking for regional adaptation or just response to different stressors. And so we identify these different really broad buckets again of like disease or pest pressures, heat, cold, drought, resilience, and then fertility. And so I won't get too far into this because I know there's another session during the summit that's specifically about this topic. But again, like we work with lots of different farmers in different places, and something we hear sometimes is like regional adaptation is like, we just say, oh, we want it to be regionally adapted. Like, oh, in the Northeast, we need it to deal with like how hot and humid the summers are. We want to just parse out what that actually means, right? Because getting a lot of rain, having a lot of water, like is plants like that. Plants need water to survive. So you want to break out like what does that heat and humidity mean, and understand like what diseases and what pests affect different crops, so that you could start to think again about what might be heritable and fixable in the long term. And so just some things to note and think about is this idea of genetics times environment. So these two different factors that equally affect, again, just what we interpret as vigor and viability. So there's some things that are just about the genetics, and those are things that we could like hopefully reinforce over these long periods of time with enough attention and sound decision making. There are a lot of things that are just environmental too, but there are some things that you just can't kind of skirt around, and you're going to continually have to like actually uh, you know be a good farmer about and just respond to what the plant needs. But as much as possible, like we want to try to, yeah, negotiate with that plant and not just, bombard it with everything that it does need, but we want to think about what levels of sort of austerity or stress will serve it in the long run. The other thing for you to just think about that we're not going to get too far into is the idea of vertical versus horizontal resistance. And so in a lot of kind of traditional breeding programs, vertical resistance just refers to this idea that a lot of traits, like you're looking at something like drought tolerance, right? And you look at, kind of like bigger multinational kind of seed companies, especially ones that are kind of housed within chemical companies, and they will find like the one or two sort of genes that control and regulate RNA transfer, which confers some amount of drought tolerance to crops like corn, right? So that's vertical because it's just thinking about like identifying the genes that kind of like control essentially those traits and tweaking those genes. Horizontal resistance is much more what anybody on a small scale and what all of our ancestors did as like traditional plant breeders, right? Which is just selecting, and there is a much more complicated matrix of factors than to achieve something like drought resistance. But what you see in places, yeah, like the Southwest, and with the incredibly elegant farming systems of like the Hopi people, where it's like you have all of these drought tolerant varieties, but it's really based on like sound agronomic systems, understandings of the landscapes, coupled with those plants, yeah, evolving in relationship to all of those things. So horizontal resistance is saying like, we may not be able to break it down to the elementary particles, but we can see a plant kind of in this rubric of different traits, and we can constantly kind of whittle it down to being optimized for the broadest amount of those environmental factors. So try to map out just the basic life cycle. And again, like this is going to focus mostly on annual plants, but most plants will follow like some level of this with different interruptants and different places. You start with a seed and you end with a seed, right? The seed germinates, goes into vegetative growth. And again, it's making all of this vegetative biomass. It's increasing its sort of photosynthetic capacity, sending all of these complicated exudates to their roots. And so all of that really is in service of equipping that plant with the ability to keep making food and perpetuating its own momentum to enter reproductive phase, going really strong. So that's the plant starts flowering, right? You get fertilization of those flowers, which hopefully means, yeah, that seed starts maturing whatever the vessel is. You have seed maturation, and then you have processing after that. So these are obviously like very big abstractions of like the life cycle, but I want to just kind of map it out because they also represent, when you're planning your seed project, what you could consider benchmarks or milestones to look at. So we're going to talk a lot now, segueing into just like the idea of selection and traditional plant breeding in terms of just like your seed keeping efforts. So, you have seed, you could do a germination test, but usually once you see it germinate, it's like you can have a timeline and then say like, okay, the first active evaluation and selection I will do is in my greenhouse once these seeds germinate. You're going to look at just like the size of the cotyledons, the general health, how consistent all of those seeds came up. So beyond just the percentage of how many seeds germinated, you want to see like, is all of these germinated seedlings like created equal? And you want to start to just, yeah, put on as much of a scrutinizing sort of microscope as possible to just observe anything that's coming through that's emergent and inspiring, or things that start to worry you in terms of like, oh, this isn't looking too good. So there you have an evaluation process: germination, and then how rapidly it moves into making its first true leaf, right? Because that food can be funneled pretty efficiently again from the parent plant into starting to photosynthesize. You could have a range of times that you're making selections then from the seedling phase. So you'd look at like rough rate of doubling, right? So how much time is passing between like making new leaf sets, how rapid is the growth, and how healthy does that growth look? Are you seeing anything that stands out in a good or bad light? So all of this that's happening over here during the plant's vegetative growth are points of selection. So for us, we start everything for the most part in our greenhouse when we're trying to make these selections at different points to just have everything that goes into the field be the strongest. So you're just trying to vet as much of the sort of things that are struggling or things that are showing signs of stress out, and go for the most vigorous members of your population at any time. And again, you're going into reproductive mode, you're looking at flowering. With crops that obviously you're eating the fruit, you're going to evaluate those fruits, right? And so you're looking at, again, all of that criteria, all of that phenotyping protocol kind of in this period. And then you're getting to the point of just, yeah, maturing the seed. You want to look sometimes at things that we're going to talk about later: ripening, post-harvest ripening, and then just general quality as you're processing the seed. So again, if you think about those things as just the times where, everybody gets busy, you get caught up in just the riptide of the season, but you want to just carve out for yourself like a general sense for each crop where those milestones land, and when you're going to be like, okay, I just know, like, I'm going to mark this on my calendar. This is the time that I go in and I put my plant breeder, glasses on, and I just view it like through that specific lens of just trying to make those selections before the next phase of that plant's life takes over. So the most basic tenets of plant breeding are the idea of selection and roguing. So one is about selecting for the things that are desirable, the things that you're saying like, hey, this plant is doing this cool thing. These members of this population are just like, expressing more vigor, just there's like a liveliness to them. There's something that you're really responding to on that visceral level. Roguing refers to just, the unfortunate act of, removing members of the population that express some sort of undesirable traits. So that could be, more susceptibility to diseases or pests. It could just be off types. So things that are like, oh, this tastes different, it looks different, like in ways that, we want to keep out of the variety we're trying to maintain. So it's just about the either like proactive or sort of, I guess it's kind of rude, but yeah, the selections that you make to decide like who's moving forward and who's not. So those are just the two acts that you want to start employing on your farm or in your garden. And that's all that plant breeding really comes down to, I guess, in a certain sense. So we want to talk a little bit about population size here, just because I think especially my big concern for me is like a lot of the communities that I work with and I feel very accountable to, the access to land is the biggest barrier, right? Like just pure, like being able to access a land base, have a relationship to land, like refoster, like heal your relationship to land. And then the relative scale that a lot of small scale farmers face: land tenure, land insecurity, things like that. And one of the things that worries me too is that the seeds really reflect that too, in terms of like a lot of the biodiversity that's being preserved is being preserved by peasants globally, right? And because of just the whole mess that we're in globally, a lot of the livelihoods and ability of those farmers and peasants to preserve that biodiversity is really impacted by the activities of countries like ours, right? So the preservation that's happening is happening in these relatively, kind of in a small scale manner, and the populations then reflect kind of like small population sizes that are able to be maintained. So even in like seed banking efforts and things like that, you still end up with a very small sampling as like crop wild relatives, and the kind of like all the wealth of genetics that exists for any sort of domesticated crop species are impacted by, just the fact that we primarily now, like a Western diet, is reliant on only 12 crop species, right? So I want to dig into population size just to not, and it makes it sound intimidating, I'll acknowledge, because it's just talking about like something that seems out of reach, just given the small footprint that a lot of us have in terms of land. But it's just something to think about because if you are really considering like, hey, this crop I can't live without, and there isn't a lot of the diversity in genetics readily accessible to me. Maybe I have one commercial cultivar. Population management and being able to grow as big a population as possible is really essential to you building back in some capability of taking a small population and steering it away from some sort of evolutionary dead end or some sort of genetic bottleneck where you start to see inbreeding depression and other traits that just accidentally start showing up and becoming perpetuated. So we're just going to go over a little bit about this because for me, the crop I couldn't live without is kidney or Korean perilla. So here are those little guys growing on my farm. So kidnip is outcrossing, but it's predominantly self-pollinizing. So for the most part, you can treat it somewhere in between something like a squash and then something like a lettuce, right? So the minimum populations that we tend to think about to maintain the vigor and health of the plant, we want to save between 25 and 45 mother plants. If we're thinking about long-term variety and genetic maintenance, like again, having a large enough genetic pool where there's possibly some recombination happening, possibly some new elements of variability happening, which is good, then we want to start thinking in the realm of 50 to 75 plants. Right, so already that's intimidating. 75 plants is like a lot of space to take up. Back. So I've got my 75 patch little kidnip plot then, right? So if I say I need 75 mother plants to think about this plant's long horizon and that my great grandkids will always have kidnip on their dinner table. If I plant 75 plants and I expect to save seeds from all of them, it leaves me no margin of error. It leaves me no room to account for losses, all those pesky gophers who also love the flavor of this plant. It doesn't leave me any buffer for the birds that really like eating the seeds, who I do want to keep well fed on my farm. And it leaves me no room for selection because all 75 of these little individual plants, right? It's like 75 people put into a room. There are going to be varying levels of plants that you want to hang out with for the long term, right? And so you've left yourself no room to actually make decisions proactively or to not save seeds from plants that aren't doing as well, right? So you end up with what's kind of like in this realm of mass selection. You just saved a whole lot of seed from a group of highly variable individuals, which again is fine if that's what has to happen. But you always want to just be trying to essentially skim the cream from the crop. I think that's kind of the clearest analogy that makes sense to people. You just will always want to be kind of leaving yourself enough room to play with that balance of how much do you have to look at and how much do you have to choose from. So we'll just go through quickly, what does this start to look like then in terms of selection? Let me just move this. So if I want 75 mother plants and then I have the space and I can plant out 105 plants, let's say, you can see each little heart represents then a plant that I've selected, like, okay, you're one of my mother plants. I'm going to save seeds from you. That gives me a selection percentile of 78%. So I'm taking about 78% of my bigger population and I'm saving seeds from that. So it gives me, that little bit of wiggle room, like 22% of wiggle room to just decide who's not coming along for the rest of this ride. And we just go through, obviously, like the math keeps changing. You double your population, you plant out 135 plants. That gives you room to say 50-50 split. 50% of you are coming with me, 50% like you're going to be food for the birds, right? And so you just start seeing how mapping that out starts to just affect your ability to, continually keep proactively moving in a direction where you're just selecting for the absolute healthiest. And the wider you can make that margin, the choosier you can be, it also allows you to grow enough where you do start to see things go wrong, and where like a stressor becomes like a really welcome part of your farm. Because if you see like a disease or something else being introduced, some sort of susceptibility, then you can hope to say that individuals that don't express that susceptibility might have something that they can pass on to future generations. So the bigger the population you have, the wider and the smaller you can make your selection percentile, the more it just means like what you're left with is like kind of the strongest of the strong. I want to talk a little bit just about the potentials for adaptation then too, as it relates to population size. To say like when we think about these ideas of regional adaptation and doing on-farm breeding and improvement, population also becomes really important because you want to be able to actually replicate trials. Like you can do things in a way and just know that like any amount of selection is going to yield something that is, yeah, more and more accustomed to your type of care, more and more acclimated to your environment. But if you really want to hone in and start saying like, I have three things, like three agronomic traits that I need to see improvement on based on the stress and the kind of pace of climate chaos where I'm growing my things, then you want to do a replicated trial. So you want to just have your one control plot and you want your experimental plot, which maybe instead of experimental plot should more accurately just be called like your hell realm plot, because it is the place where you see a lot of struggling and it will just be hard to witness, but it's in service again of that long term. So again, you want to just be able to have large enough populations where you can safely say that there's some correlation that you're seeing in the interaction of how you're treating those plants and what they're responding to and the stress that they're enduring, to having something that you can track heritable outcomes over time. And so in your control plot, let's say I was looking like, oh, this perlay is great, but it needs a lot of water. That's becoming increasingly scarce, and its fertility needs are somewhat moderate. So I want to design an experiment to see, can it use less water? Can I get it to thrive with like no fertility? So rather than just throw everybody into that mix and everybody has a horrible season, you develop this sort of two plot system to just see, again, what's your baseline and how far below that baseline can you bring things where enough of the population struggles and doesn't do well, but a certain percentage does do well. So in your experimental plot, you do want to see maybe up to 50% of that population like struggling, really, really being stifled. You don't want just sheer complete morbidity, but you want to see enough that like there's a difference, right? Because if there is no difference and everything just looks fine, it's like, what you can assume is, oh, I've been giving everything too much water and too much fertility because look, they're really unfazed and I could just drop it down there. You adjust that baseline. If you start seeing like a real, kind of smattering of different responses to your applied stress, then you start selecting for those individuals. And so we did this with drought tolerance with a type of heirloom eggplant, just because again, yeah, it's known as like this really thirsty, greedy crop, this type of, really particular Japanese eggplant. And so we did this over a series of 5 years. And with each of those years, your treatment might be the same, right? Your control plot maybe always gets a certain fixed amount of hours of irrigation. You're giving it compost or whatever your fertility regimen is on your farm. Your experimental plot, you just decide what is the ratio and how does that relate to your control plot. So with our eggplant, we just gave it half the amount of water each week and we gave it no fertility. So we threw it in the deep end in terms of the fertility needs, but we still irrigated it because it seemed like pure dry farming was out of the question. So you just, again, develop that stress threshold. And it's only after a few years that you start seeing any progress that you can say like, okay, some of these things are actually becoming heritable. And so after about 3 years, our two plots started looking more and more like each other. So that gap starts closing, even though the treatment is staying fixed. And so those are the things that you want to map out over a couple years. What's the timeline I have to figure this out? And if you just see like year after year, that plot looks completely miserable, you might not be making any actual advances, but you might still have a couple star people that are figuring things out. But if it's not heritable, you don't know what to account for too. Like maybe there was an irrigation leak somewhere. Maybe there was, like one pocket of especially wormy, beautiful soil, or something is happening in terms of just the the soils we're growing in aren't completely uniform, right? So again, it's not to make it intimidating, but saying as big of populations as you can grow over as long periods of time to like really put your scientist hat on and try to really track this information in some structured way just means like in as little as 5 years, you could have something that is like really, really optimized for your farm. And I think that's a really big deal. Just, I think it was surprising even for me that you could have something that does seem to be tracking in this kind of inspiring progress in under a decade. So now we're just going to go into a little bit of just the processing itself. And this is the part that, yeah, again, it's a bummer to not do this in person altogether because it is really fun. So dry seed processing, we're just going to talk about first. So this is obviously, seed that's held within a pod, a capsule, or seed head. So things where that dry down process is just kind of happening naturally. It's not inside a fleshy fruit, right? So your different types of greens, herbs, um, yeah, like, and there's some things like chili pepper where it's somewhere in between where there's debate about processing the seed fresh from the fresh fruit versus like drying the fruit first. But this is mostly, yeah, like a lot of the things that, brassicas, lettuces, herbs, things like that. So the process is, from flowering and reproductive maturity, you have that flower being fertilized. You have the seed maturing within whatever the sort of ovule is. The seed coat then develops after the embryo is actually plump and sized up. And then you have the plant has fulfilled its mission, right? So it has no more need necessarily to be making more leaves and things like that. So the plant will start to dry down as part of like kind of the finishing process with the seed. The activities you tend to do with dry seed processing, threshing, whacking it with a stick, hitting it against a tarp, hitting it inside a bucket. So just the forceful, removal of the seed from whatever is containing it. Then you will use probably some form of screen, and then you will winnow, which is using some sort of wind to kind of take out the lighter chaff. So again, we're going to keep with our perilla as the sort of example we can use. So on the left here is our genip, and you can see at the sort of terminal growth points, when it's in its vegetative mode, you just see new leaf sets in here. They look like these other leaf sets, just smaller. And perilla is something that has like what's called like an obligate photoperiod day length sensitivity. And so it really is like it will not go into any sort of flowering mode until like its photo period is hit. And so for us, that's usually around late August or September. And once it goes into that reproductive mode, it ceases to make leaves. So as opposed to like basil or something where if it starts flowering, you could prune it back hard and find a node where there's some axial buds that are still putting out vegetative growth. All crops in the perilla genus, once it's decided like it's time to flower, you could prune it back and it's just going to make flower racemes from like any little node that it can. And so there's no point to trying to prolong it or trying to divert it from flowering once it kind of enters that mode. And it also doesn't matter. It's why it doesn't make sense to succession plant it, because if you plant it too late in the season and you're close to when it's going to start initiating flowering, it'll do it regardless of how big it is. So on the right-hand side, you see once that flowering is initiated at its apical meristem, it's not making any more leaf growth. Those little pieces are the beginnings of the flower raceme development. So those are all actually tiny sepals now instead of new leaf sets. And so now you see those leaves, the sepals starting to expand, and the space in between them is where those flowers are going to develop along that central raceme. And so here you have it kind of at like right after its peak flowering. And so all of these kind of along that stalk are the little calyxes left over. You see the little white flowers up here that have bloomed and dropped. Um, and so all of this, like they start flowering from the bottom up. So from the bottom up of the plant and from the bottom up of each individual raceme. And so it blooms in succession. So you have the less mature seed up top where you can still see the petal holding on. It's just been fertilized. And then towards the bottom, you can see the dried petals here. And then you see the swelling kind of at the base of that calyx right in there. And so this is just the cues that you're looking for, because in terms of like post-fertilization, what you're trying to track is like how the seed is actually developing, the pace at which it's developing, and if that's staggered kind of like at different places along the plant. Because when you decide it's the time to harvest it, you're looking for both like, you know, getting the highest yield from the plant, capturing the most seeds before they drop or are eaten, but you also want to kind of cut it in the middle where you don't want necessarily just the first seeds that develop. You want to kind of stagger it a little. So it's like, you know, maybe after that first 15% mature, you want to capture kind of like the prime 85% of seeds, if that makes sense. So for that, you're looking at a lot of visual cues, and the things we really rely on is like, we know peak flowering because you just hear it. You hear like the perilla patch is like completely buzzing with bees at that time. Pollinators love it. Once that starts to subside, then we start looking kind of every day at just how these little seed capsules are developing. And then you can see there's a beautiful language that a lot of plants express, right? Because they have brought us in as these conspirators, right? So you think of like a fruit being like so seductive in terms of like you eating it and helping them disperse those seeds when it's mature. Even your dry seeded plants where you're not necessarily eating the fruit, like have these expressions. And so here you can see, you know, if you look at the top, once those flowers drop, the calyx kind of closes like a little, say like a little kissy face almost, like it kind of closes up because it's going to protect the little embryo developing within. As you start moving down the raceme and seeing gradually more and more mature seed, you see again that little calyx slowly opening back up, right? Because it's getting ready for seed dispersal, then it doesn't want to hold so tightly to it. You see the swelling at the base as that seed is just physically getting larger and larger. So by the time it's mature, you can look inside here and you could see the 4 little seeds inside that little nutlet. And so again, it's just trying to tune into like the cues, because the plant is really wanting to enlist you as a helper. So you start looking at those little beautiful kind of like body language, essentially, of just taking your cues from them of like, okay, you're showing me you're getting ready. You want something to come take this seed pretty soon. That white seed means it's sort of, you can say like the embryo is hit maturity, but then you're waiting for seed coat development. With a lot of dry seeded crops, you're the main thing you're competing with a lot of times is either like shattering and the seed dropping, or honestly just a lot of predation from other animals, right? So once it's queuing you up, you can assume other species are also tuned into it and ready to take those seeds as well. So for us, with a lot of our dry seeded crops, once we know the embryo is mature, even if the seed coat isn't developed, we can just know for different crops like when it's okay to cut it, bring it indoors, and let it finish drying down. So you want to just figure out like when the seed is actually mature and when that post-harvest process can happen in terms of drying it down. So we cut it, we bring it inside into like our greenhouse, which is covered with a heavy tarp to just make it really shady and let it dry down kind of nice and slow. Because again, you're not trying to roast it or singe it. You just want the seed coat to continue developing. And yeah, the great thing is with a lot of crops, like it's continuing to direct a lot of energy. And right now, all you're trying to do is lay the plants out. The key things here, especially if you're in humid climates, is just air circulation. And so we lay this out kind of just banquet style. Because we're in such a hot, dry climate, we get allowed to be a little funky like this, where we can pile plants a little bit more than I think people in, practically any other part of the country can do. But essentially, you're just laying them out. You want to cut as close to the base as possible, but it doesn't seem, I know a lot of people like to try to pull out some of the roots too, but it doesn't totally seem necessary in terms of just like, if you keep this sort of like vascular cambium intact, it's still directing a lot of kind of metabolites to the maturing seed. So you're letting it dry down. And this is the phase. Now you look inside that little calyx and the seed is turned dark gray. And all that's really happened in this process is drying and seed coat development. And so then we put it through a process of screens. And so basically the fundamental thing with screening in this part of the cleaning process is you have one size screen that you're putting all your mats into. So you, I guess we'll go back. So once it's dried like this, you could pick up the plant and shake it a little and you'll hear like a little rattle, right? Then you can think, okay, this is probably pretty ready. And you can experiment with the timing of this to just see when your ideal window is, and that'll just vary from farm to farm. But once you can kind of like pick it up, it seems fairly dry. Threshing is very easy. You lay out a tarp, you try to whack it with a stick, and a lot of the things are going to want to fall out, right? The seed is going to want to disperse. And again, this is like kind of the sort of law of diminishing returns, or just leaving yourself as big of a runway as possible, because at all these stages you're going to be constantly skimming and your overall amount is getting smaller and smaller, but that is just the good, what you want to see at every stage to keep ensuring like quality maintenance. But again, so here, you know, you whack it, take everything that falls out. You don't want to spend a lot of time, like you may just get obsessed, like, oh, each raceme, like there's seeds that are still in here, trying to grind it up in your hand, trying to really kind of remove every single seed that the plant has made. You really want to stay away from that, because you want to just take the things that, again, that are heaviest, that are most ready to come out, that the plant has really said, like, hey, you're ready to go out there. I'm ready to disperse you. So again, you know, trying to save more than you need, so that you end up with the amount of like physical seed at the end of all of this processes that's close to what you want. But you don't want to be afraid of leaving some things in the plant or having some things just get winnowed out in the process. All of those are good things in terms of just showing that you're having the best of the best. So after you thresh everything, you have all this plant matter, you have seeds mixed with some of the leaves and the dried up racemes. You first screen it through a screen where the screen size is just big enough that your seed drops through and a lot of this bulky biomass stays on top. So this is our first screen that's just big enough. Our little perilla seeds drop through. All the leaves, the rest of these little flowers all stay on top. Your second screen catches your seed, which you see here, catches some of the finer chaff, but a lot of like the dust, maybe some tiny little bugs, some of that finer material passes through there. So now your screen is just a little bit too small for the seed to fall through, but everything else. So you're just trying to cut it in as narrow a margin as possible with too big and too small, so that it's as well sized to catch in the middle the seed as possible. With Perilla and with other things, you may want to grade the seed too into like 2 different lots. And so sometimes with Perilla, because it is also a really valuable food crop, we take that first screen of the biggest seeds and we reserve that for planting. And if the smaller screen, which is just the next size down, catches some of the smaller seeds, we end up screening that and using that as food. And so if you want to start separating things out too, to just kind of like see the percentage of like what that size differential is and how much big seed you're getting, you might want to just do like a 3 screen process so that you just start to see some of like a quality indicator, so to speak. The next day after this is winnowing. So you obviously could use the wind. We'll use like a series of small fans just to kind of like, yeah, really dial everything in. So on the left side here, this is our cleaned Perilla seed. So nice and gray, all consistent size. On the right is what's left on the ground after we're fanning. So you can see maybe slightly different size, but just that the seed is lighter, the seed coat isn't as developed. And so it's seed that just was maturing later in the season once it was cut, wasn't at the phase that it was, that these guys were. So this is kind of like more mature, kind of heavier and more vigorous than these. So again, it's like with winnowing, you feel very precious. You're, oh no, these seeds are flying out, trying to conserve that. But as long as you're not like, yeah, really blowing things out like wild, it is always good to see again, like some things flying out. And you want to kind of like, as you're pouring your seeds into the fan, you want to just see like where those seeds are falling and how much space between the vessel that's catching them with what's being pushed out or pushed into a secondary vessel. And just kind of gauge that where you want the seed to be landing fairly close to the edge, so that things that are just like a touch lighter don't make it into the vessel, so to speak. So the main things you're thinking about when you're fanning are just, yeah, the speed of that wind, and that'll just depend on like the size of your seed. So like, something like mugwort seed or yarrow seed is so incredibly light. So you're going to just put it through probably a lower speed than something like, beans or cucumber seed and things like that. And so, yeah, you want to think about the speed of the fan, the distance that the seed is dropping overall, and how you kind of adjust for that based on how you see the seed being captured. Kind of how high or low you hold your container so that the seed is falling before it hits that turbulent sort of strip of air. So those are all things that, again, it honestly just unfortunately comes down to just kind of muscle memory and practice. So wet seed processing really quickly, seed obviously held in a fleshy fruit. You have like, once it's in reproductive phase, you have fertilization, the maturation of that seed. And then in parentheses say, because it is really important, but it's there's just varying ideas and based on crop species, like the importance of this, but post-harvest ripening. And then the processes that we do to just clean and store the seed is you're usually fermenting it in some regard, because a lot of things that are held in some sort of wet, fleshy fruit need to have some mechanism that's inhibiting germination so that they're not just sprouting, right? Because if it's just like inundated with water inside of something like a tomato, you need something that is telling that seed like, no, it's not your time to grow. You need to be saved for the future. Then you're washing and drying. So we're going to use our chame, Korean melons, as an example for this. So this is just showing, on the left here, that is clearly, melons, they have different flowers for producing pollen and different flowers for maturing seed, right? So stamen and pistillate flowers. So we have here a flower that is clearly attached to what could evolve into a melon. So the ovule is right at the base. You have that flower blooming and opening up, ready to receive pollen. And then you have it just being pollinated, and the showy petals obviously aren't necessary anymore, so those will drop, right? It's important to kind of note this phase, and if you want to get into more detail, like a really good thing to look at is what's called days from anthesis, and that is days from this kind of like prime flowering, when the flower is totally open, that kind of when it's essentially going to be fertilized. And if you look at different things, if you want to really geek out about different crops, like you can see different results in terms of like just seed heaviness, seed count, like long-term kind of like health outcomes for that growing plant. And days from anthesis is like a really key indicator of like when you harvest the fruit then. So you want to just kind of always be aware, like, is this kind of this peak, how many days out? So that affects like the maturity of your fruit, right? And that's just saying like, don't, like the fruit needs to be as mature and possibly over mature in a lot of species for the seed to actually develop. So for a lot of things like, cucumber, we're mostly eating the immature fruit. You need the fruit to go like way above and beyond that to harvest the mature seed. With melons, it's a little more subtle. So you have here, you can see we have that fertilized flower, that ovule starting to develop into a cute baby melon. You have that melon kind of in pretty close to harvest, right? The fruit is set, and then it's just going to be, subject to some ripening. Then you have the fully ripe melon here. So the color is changed. Once we're at that point with melons, like you end up with like a better chance at good quality seed, even when you harvest at the stage that you're eating it. So lucky for us, melons are good sports in that regard. Post-harvest ripening. So most of the wet seed processing we do, regardless of crop, we do have some period of post-harvest ripening. So with our tame, we pull them off the vines once they are just slightly over mature, and then we keep them in like a cool place where the temperature doesn't fluctuate too much. It doesn't need to be a cooler necessarily, but just somewhere where there's not a ton of variation. And we'll store it there for, if we can, between 10 days and 2 weeks. And especially with a lot of cucurbits, the post-harvest ripening actually like really influences, because if you especially leave some of the stem on, there is the ability for that fruit to continually channel like any of the leftover photosynthate that it's accumulated throughout its life into seed and direct it into filling a higher quantity of the seed, right? So, within that one melon, let's say like you harvest that fruit and about 70% of the seed has been like, kind of embryologically mature at that point. If there is lighter seed within it, that post-harvest ripening helps direct more photosynthate and metabolites into the unfilled seed. So you can kind of slightly up that percentage to maybe get, like in the mid 80s then of like filled seed by the time you're done with it. There's a lot of other ideas with, so with a lot of the fruiting plants, there's some element of indeterminate growth where it will simultaneously grow vegetatively, right? So a lot of crops like tomatoes, cucumbers, melons, all of squash, it'll start flowering, it'll produce flushes of fruit, and then it'll alternate again. It'll start to build more vegetative biomass for itself, do another round of fruiting. And just to say there's conflict of opinion with people in terms of, again, like you want to flag your plants, like any dry, wet seed processing, any plants that you're saving seed from, you really want to like decide that's a mother plant. You want to really center all your decisions around how you harvest and how you treat that plant by determining who are your little seed plants partway through the season. So that goes back to that sense of milestones. You're going to put a flag or just mark each plant that you see as your little star people. You want to just kind of keep that in mind. And so there are some decisions that you may end up making in terms of just, like the ways that you harvest or don't harvest, or thinning fruit or pruning plants that will dictate like directing. You're just trying to have the most amount of energy directed to the seed as possible. And so like with our dry seeded, like with perilla, the plants that we flagged as our mother plants, as we know their flowering is about to begin, we hold back on harvesting the leaves because we actually want like it to retain as much of its biomass, as much as those little solar panels as possible, so that it's creating as much food as possible. So we leave those plants alone and we'll harvest from the plants that we're not harvesting feed from, the leaves up until they're done. With melons, for example, there are some lines of thinking that say like, you want to see that flush of fruit, and then you want to thin it, right? So the same with a lot of fruit trees, you thin off fruit to get less quantity, but maybe better quality. So I think there's a thinking that's similar to that, where especially with vining plants, also on your mother plants, you may want to prune the vines to just where the fruit is. Because again, like you're creating a sort of terminal point that ends with the fruit. So hypothetically, like all of the nutrition and things accumulated by that plant are going to just be directed to less fruit, more mature seed. We don't do that just because of time and laziness, I suppose, and also just unnecessary pruning. But it's again, like if you just feel like you want to just really maximize it or something feels very high stakes, you might want to consider some of those management decisions. We post-harvest ripening. After that, we scoop the seeds out with some of the flesh. We cover it with a certain amount of water. You want to just add enough water so that you can see that separation. So, right, like if I had just scooped out all the flesh on the seeds, I have this dense mat of just fleshy matter. It's harder for the seeds to kind of like drop in and out of, and it's harder for me to see that separation. Um, so here you can kind of clearly see it separating. Your viable seeds fall to the bottom. They're heavy, right? And so you really immediately also get a sense of like kind of a germination, like a rough germination estimate. The rest of this, you leave fermenting. It can vary from, like if it's super hot, you may only need like 3 to 5 days. If you're in a cooler climate, you may need like a week or more. But you want to see some fermenting. You want it to smell fermented, but you mostly, at the end of the day, like, you'll pull out some of the seed. If you still can sometimes physically see like a film around the seed, then it's like you may want to subject it to some more fermenting because you're trying to break down a little bit of that, again, germination inhibitor. But from here, you have, this cloudy water, which is good too, like for feeding your plants, in terms of just like cultivating a lot of your native yeast and just being like a really nice kind of electrolyte plant Gatorade, I guess. So, that's also useful. Then you're just going to decant it. So you have these jars, you have your seed at the bottom. You first kind of pour off, shake up the top layer, see if anything else wants to fall down. You continually pour water in. Your heavy seeds are going to stay at the bottom, and you're just trying to loosen up and get anything else, like any other biomass that you don't want being stored in the seed that could open it up to mold or something else in the long-term storage. You want to just skim that off. And so with each subsequent wash of water, I would pour off, like all of this. I'd pour in more water of that same amount, let that separation continue. Eventually, the water that I pour in and swish around, once that's clear, you're good. You pour that out, pour this into some sort of sieve, and rinse the seeds. And then from there, you just have to figure out drying it really well. So for us, I like just using basic ceramic plate. We used screens. I didn't like the metal being kind of like up against the seeds a lot. You could use some sort of parchment paper. We use reame or row cover sometimes for different bigger seeds that helps wick away some of the moisture. I've heard people using fabric tulle for smaller seeds like tomatoes that just get clumped together, because if you use a paper plate or a paper towel with tomato seed, it's like you're just going to have it's going to be dressed in a little paper towel outfit for the rest of its life. So yeah, ceramic plate, something like that. And so you could see like the wet seed here is kind of like that nice cream color. And then as it gradually dries, it'll get more matte and white. So again, you just want to keep this. We don't leave it out in the sun. We keep it in a nice shady place where there's a lot of air circulation, nothing too stagnant. You stir it around just so that it kind of dries as evenly as possible and nothing's just like getting shellacked onto the base of whatever you're drying it on. And that's basically that. There's your finished melon seeds. So just want to close by saying, like the other thing to just consider is like, I just encourage everyone, make it as collective as possible. You could get caught up in just like all your own preferences and things like that. But for us at the farm, the real joy of all of this is like inviting in as much participation as possible. So when we evaluate anything, any breeding project, anything we're trying to stabilize, we want it to be a reflection of our community. We want like every bite we take of all of these fruits to like bring us back to these moments that we shared with the people we love. And we just think that's a really important thing because it's what also it feels like those seeds and those plants are asking for. It's asking for like you to build a team around them of people that just feel very devoted to ensuring their kind of longevity, to holding them in these kind of like webs of care and reciprocity. And it also deepens your relationship with your community, like to just realize like there are so many acts of gatekeeping that have happened in the seed industry and the seed world that we look and we feel like we've been dispossessed of the kind of like most primary task of agriculture and the thing that unites all of us in terms of like what all of our ancestors did. And the beauty of that and how it shaped like the foods that are these links to one another. So again, I would just emphasize, like, make it as community based as possible, like to whatever extent that feels feasible. Also, then you have, children enjoying things, like building these habits and like really learning to relate to these crops in such a different way. We've been able to really not mechanize to a large extent, regardless of the kind of scale of seed processing we're doing, because we have people that enthusiastically just love stomping on beans and things like that. We won't ever need to buy a combine. Um, and yeah, again, just kind of like taking opportunities, so that I think it's important for me as a farmer, for like people who aren't farmers, people who just think I don't have a green thumb, I kill everything I grow, but to still think of there being on ramps for them in terms of like this process. And if seed keeping is more than, yeah, the act of removing a seed or like carrying a seed from season to season, what are the different roles that everyone gets to play in that process? Because we need storytellers, we need cooks, we need people that just love to eat. We need people that just, yeah, want to come and spend time with those plants. And so, yeah, that to me is just the sentiment I wanted to leave everyone with, is just thinking about our collective responsibility, and not seeing it again as like this technical task that farmers even feel kind of has been taken away from them, but that a lot of communities have felt like, uh, yeah, really unfortunately kind of robbed of for a long period of time. Um, so that I will end my presentation and stop the sharing. I know it's kind of time for discussion, but you definitely want people to chime in and share anything that feels relevant to you all, or also have time for questions that people want to ask. The Chatmeister left, I think. But feel free to, anyone can take themselves off mute if there's like something that, a question that anybody wants to ask. Okay, someone raised your hand. Yeah, go ahead. Feel free. Just, I think you can take yourself off mute. No, cannot. Okay, let's see. Okay, it says I'm asking you to unmute. Can you do it? Okay.
Okay. Hi. I was wondering, with some of the other presentations, they have said that there's going to be an email come out with a link to this presentation that will be available for a couple weeks, along with like a documents folder. Is that going to be available to us for this presentation?
Yeah, it's being recorded now, and it'll be uploaded to wherever the planning team is putting all of this.
Thank you.
Yeah, of course. Okay, Stephanie Berganini, I ask you to unmute. Can you do it? Okay.
I think I did it.
Okay.
Thank you. First of all, just thank you. This presentation has been amazing. But I wanted to summarize a couple of questions I saw getting asked in the chat a lot. Which is just, is a like heavier or bigger seed always better? Is that sort of a universal piece of advice about seed saving, or does that vary?
Let's see, I'm sure it can vary, but I feel like it's, yeah, even with really small seed, and there might be like other more nuanced attributes to keep in mind, but in general, like, yeah, heavy and larger just tends to be kind of like what people have always like. It accounts also for like those kind of big impact mutations throughout time, where people just wanted like, yeah, larger grain and things like that. So I mean, I would always avoid like saying anything absolute, but I think definitely in terms of heavier, that's going to be really consistent, just because, yeah, it's just kind of like filled out in a different way. So yeah, I think for the most part. Okay, I see Laura. I ask you to unmute.
Hi, Kristen. So my question is about the fermentation process. Do you always have to ferment seeds? And also the medium on which you dry them, can you do it on like a tea towel? Somebody else mentioned coffee filters. I've always gotten paper towels stuck to mine and just planted them in like that. Maybe that's a problem.
No, I don't think it's a problem. Yeah, I think so with wet seed processing, let's see. So something like bitter melon, like if you've seen, like the very hard brown seed is encased in like a red gel called an oral. Um, uh, and that doesn't necessarily be fermented, but you're usually loosening it up in some way using water, so that you do need to have some subsequent drying. I know there's even a huge difference between something like like a tomato with something like a melon, where the germination inhibition isn't as strong. And then there's things like that are technically like wet seed, like winter squash, or yeah, any sort of squash, cucurbita species, where you're not fermenting it, you're just maybe washing it and then drying it. So yeah, not all wet seeds have to be fermented. It's just knowing if there are some that have some sort of inhibitor. So yeah, even the difference, like tomatoes really do need that, whereas eggplants don't, peppers don't necessarily. But in some ways, like depending on like the fleshiness and like the nature of the fruit too, the fermenting just also helps clean off the biomass more than like regulating any sort of germination issue. And then yeah, I think what you said, like a tea towel would definitely work, coffee filters. Yeah, I'm always entertained because like fabric pool has come up on a number of things as like a cheaper form of like insect netting. It was like, oh, you go to like a farm supply and insect netting is like just wildly expensive. Then you could go to a fabric store and fabric pool is like a lot more affordable. So I think, yeah, that could work as well. Okay, Lauren,
Thank you.
ask Lauren to unmute. There we go.
Hi, Kristen. Thanks for a great presentation. I learned a lot. I had a question for you. So what at times are your highest priorities for seed saving? Is it preserving biodiversity, increasing seed stock, adapting to climate, or does that just depend on your growing conditions? Or, I mean, are they same for you or do they differ?
I think it varies from crop to crop, I guess, but it's a really good question because again, it's like you come up with this like weird overlapping Venn diagram, I guess, or just like like a kind of weird matrix of priorities. I think in a lot of regards, like when I was learning, it was simultaneously like, yeah, having crops that were like specific to Korean communities, and then but being in a climate so completely different from Korea, where it's like kind of opposite. And so it was hand in hand, just out of necessity. Like if I want to keep growing these, they're going to have to deal with this extraordinary drought stress and not being somewhere with like, yeah, renewing rains throughout the entire summer. And most of our work, because I think originally, like with my farm too, like a lot of our focus was on like natural farming methods and kind of agroecology. And so there's always that lens. And I guess that's the one kind of constant, is like kind of always selecting for kind of low input needs and the ability to be responsive to less applications of water. So that's sort of like our baseline. Like anything, just even in terms of practicality, if we're going to keep growing, it has to be like movable in that way. And then a lot of the other work, yeah, it's focused on like kind of culturally significant, sort of like interesting traits. And then more and more just even, yeah, just being curious too about doing a lot of trials to try to preserve each kind of just potential parent material. And I think that that's something we really enjoy too, is a lot of trials when I was learning and doing trials for seed companies was focused on finding like what is the best. And now I kind of appreciate doing trials to think like, what makes each of you special? And, like, I think because again, like we just are really running out of just even like the things available to us. And so I think it's more and more important to trial things with like a broader scope of just trying to be like, you're here for a reason. You've stuck around for centuries. Someone had to like you. Like you had to be someone's favorite. What is it about you? Instead of just being like, well, you're not my favorite, so hit the road, you know? But yeah, I mean, I'm curious too, like if, yeah, it feels like there's like one primary thing for you that is like the utmost importance. Like, is it, because for some people, it's like, I'm focused on regional adaptation. A lot of our farmers are focused on cultural preservation. And so, yeah, curious if folks feel like they already have that in mind. If you, Lauren, have that, like, oh, my main focus has been this, but I'm curious about these other things. Oh,
Yeah, I'm so new at this, but what drew me into it was cultural preservation, something that was connected to place and people. And then also for climate adaptation or local or regional vigor.
mhm. Yeah, I think that makes a lot of sense. For me, it's like just being kind of like a good citizen on like not my ancestral land. It's like, oh, things have to integrate. You've got to like be here and be like a good member of this like ecosystem and landscape. Other, you know, like bad actors, you gotta, you know, leave behind or have them grow somewhere else. I know Carrie. Okay, let me ask you to unmute.
Thank you. Yeah, sorry, I don't know. I wasn't able to raise my hand, and I know we're getting close to running out of time, but I was wondering what the timeline was for fermentation and washing, like just a rough, like, does it take a month? Does it take a week? Is it within hours? Like, what do you know?
Yeah, it really varies, right? Like, so if we do kind of prime tomato seed fermenting, like in our temperatures, like last year, things fermented in like 2 or 3 days because it was
Yeah.
like 115 degrees by the time we were fermenting it. So it's hard to say because it really like just depends on your situation. Even for us, like we don't have a lot of hard and fast rules because year to year, it's like the temperatures between May and October are all over the place.
Mhm.
But yeah, like if you're doing things and it's like a late season crop and you're closer in the fall, like something like a tomato, which tends to have a more a longer need for that fermentation just generally, you know, it'd probably take between a week and like 12 days would
So you're
be
just
safe.
kind of looking for like, how does it look? I guess, you're kind of, you have to just kind of gauge it and.
Yeah, exactly. And you'll
figure
know
out.
if you push it too far because the seed will start to swell. It's going to start like imbibing that water. You'll see it starting to sprout or like mold, undesirable mold. Like you'll see some mold maybe forming on the top. And you just skim it off and it's
Mm.
fine. But really you're just kind of looking at like you can see on the seed itself, like, you know, a tiny little sort of encasing. And you're just kind of trying to time it with that. But again, it's unfortunate, like I'm sorry to say, it's like just real is some trial and error
Yeah.
and seeing each crop kind of because each crop in each climate, even in each little tiny household and neighborhood will like behave differently.
Sure. Thank you.
So sorry, not super specific, but
Yeah, no, that helps. Thank you.
okay. And I see just one more thing about best way to store seeds, which is just you know, cool and dry. Again, it doesn't need to be super cold, and I would like kind of discourage like things like refrigerating and freezing just because there's a little bit of cell wall damage that ends up happening like in those sort of like fluctuations of just like unless you have something that's like really dialed in, like, you know, thermostat controlled specifically for seeds, a lot of it can kind of damage long-term storage if it's just like in a sort of household refrigerator or freezer. But yeah, you want to just remove the things that start like having it expend any sort of respiration, any sort of metabolic activity. So you don't want it to be queued up by sunlight. So you want it to be somewhere that's dark for the most part. Um, yeah, again, like things that aren't overly humid. And then places where it just, the temperature doesn't fluctuate a ton. So people just think of it like, I don't really drink wine or know about wine, but people say it's like the way you store wine is like the way you could store seeds without getting into some like, yeah, really heavy infrastructure just centered around it. And then, yeah, we store it in, paper packets. We store them in like clay jars. We store them, some things that have a really short viability, like so where the oil content is really high, like rosemary seeds, perilla seeds. Um, we do put them in kind of like vacuum sealed bags. And with larger things like beans, if you have like some sort of vacuum sealer, it's good because it also just inhibits like if there's like any little weevil or anything that's gotten in there. And I'd say the other thing, like if you are, yeah, you have like a pretty expansive seed collection, like to break them up and store them in little lots so that like, yeah, if there is the introduction of a pest or something, like you just kind of spread out the odds of it just decimating like an entire seed lot. Yes, all the contact info, everything else will be available, I think, to all participants. And so, but thank you everybody for hanging out on a Saturday morning and